All right, in this lesson, we are talking about adaptive immunity. Here are our 2 dot points. Now, they might not look like much, but they are hiding a lot of very important and complex information. All right, let's review the immune response. We have previously spoken about the innate or natural immunity. We are now talking about the adaptive or acquired immunity. This is a kind of immunity or immune response that is trainable in the body, okay? All right, a reminder, we are going to be talking a lot about antigens, and these are substances which trigger an immune response. They can be triggered by self, but usually not, okay? Pathogens are those molecules or organisms which cause disease. All right, quite a lot going on here, but remember, in order for a patient to, or a person to recover from a disease, they need to use both the innate and adaptive immune responses. Okay, adaptive or specific immunity comes in 2 parts. There is the humoral and the cell-mediated. And these responses, operate together in a specific immunity to fight infection and retain a memory of this infection as well, so that down the track, a new response can be mounted really, really quickly. The humoral response uses B cells. The cell-mediated response uses T cells. Okay, these are lymphocytes. These are types of white blood cells. I will refer to them as B and T cells. These are cells that we talk about in white blood cells, but we're talking here, we've previously spoken about these non-specific ones, so these are macrophages and neutrophils, whereas we are now referring to quite a number of other types, specifically the T cells and B cells already mentioned. They all have varying and complex roles in a very large and collaborative response. All right, T cells and B cells both originate in the bone marrow, but T cells mature in the thymus, hence their name, and B cells mature in the bone marrow, okay? They tend to reside, they live around the lymphatic tissue and the lymph organs, so like the spleen and the lymph nodes. You'll often find when you're sick, your lymph nodes and your neck swell up, sometimes under your arms as well. And if we are comparing these responses, the innate response, we're talking macrophages, that can be a really quick response, but when we are talking B and T cell responses, we are talking much longer responses. All right, let's talk specifically about the humoral response, and that is all to do with B lymphocytes. They manage this response. And these B cells have antigen receptors on their cell surface, okay? So they can actually pick up things like those antigens that are going, okay, that's not a self cell. What am I going to do with this? Once they bind to this antigen, these cell receptors on the outside, the B cell becomes primed and it produces large numbers of things called antibodies, right? Antibodies are protein molecules, okay, and they have specificity to a particular antigen. They come in these weird shapes, often T or Y shapes, or these beautiful star shapes of IgM. Ig stands for immunoglobulin, which you probably would have heard before, and essentially all antibodies are the same except for these little antigen binding sites at the end of the little arms, okay, and these binding sites attach like a lock and key mechanism in the specificity, sort of like enzymes do. Now, antibodies, again, come in all shapes and sizes, but the B cells can only produce one specific type of these antibodies, okay? And we can produce about 10 million types of antibodies, all of which react against non-self antigens there. So the specialization of the B cells occurs in the bone marrow, and this is called clonal selection. It's essentially where they take one B cell stem cell and they go, okay, we need to be able to have antibodies against this antigen, that antigen, and the other antigen. So we need to actually sort of evolve them in that way so that they can make all those certain types of antibodies. And so they are, the clonal cells make copies of their specific antibody and they embed them in their little cell membranes so that while they're on the lookout and circulating, in case they see that matching antigen, then they can be activated. Now, the humoral response is a little bit simpler than, say, the cell-mediated response. And the antibodies that are out circulating have multiple attachment sites. So they kind of hold on to 2 different bits of antigens and they can then clump and form this antibody-antigen complex. The antibodies are free-floating, so they're floating around the blood and they bind to all these antigens, and it leads to so many different outcomes, okay? They can kick off the complement cascade that we talked about in the innate immunity. They can tag pathogens for removal by those phagocytes, again, in the innate immunity. They can prevent toxic antigens, so actual toxic pathogens, from causing cellular damage, and they can cause the clumping of those pathogens so that they are immobile and they can't spread as easily, and also those phagocytes can come along and engulf them. So there's a lot happening here, but you can see these are all things we just talked about. So we talked about the antigen-antibody complex kicking off the complement. They also clump those together, so the agglutination and the precipitation, as well as the neutralizing of toxic pathogens there. And once that happens, it sort of then kicks off some of those other processes to come in, often the ones we associate, like phagocytosis, with the innate immune system. Yep, I just said that. All right, so once a circulating B cell meets its matching antigen, it activates that cell and it can form two types of cells, plasma cells or memory cells. Now, the plasma cells produce huge numbers of the matching antibody, okay, so they can circulate for a long period of time as well. But they also make these little memory cells. Now, much fewer numbers here, but these cells are able to recall, embed those antibodies in their membrane and circulate for months to decades, so that if you meet that specific pathogen again, it is ready to go. So the antibodies can circulate for about 28 days, but these memory cells can circulate for those years, and they're not secreting any antibodies, but they have them in their membrane. So if reinfection actually reoccurs, the response can be mounted really quickly, and therefore you can clear the pathogen sometimes before you even know it. So hence why you may only experience chickenpox once, and then your body will clear it if it ever meets it again, because it knows what to do. So if we talk about a first exposure to a pathogen, obviously the antibody numbers are increasing, but then it will clear the infection, and the second time the antibodies can come out faster and in greater numbers. So that's where we'll pause for that, remembering we are talking about the adaptive immunity or immune response, and therefore it is a trainable response.